In last some videos, we have started with the types of enzyme inhibition, and we know that enzyme inhibition, they are of two types, reversible inhibition and irreversible inhibition. We have already finished about the reversible inhibition, which is of three types, competitive inhibition, uncompetitive inhibition and non-competitive inhibition. Now we will discuss the next type of inhibition, that is the reversible inhibition. Now, what do you mean by irreversible inhibition? In the inhibitor, here in irreversible inhibition, as per its name, once the inhibitor attach to the active site of the enzyme, then that process cannot get reversed and enzyme loses its activity forever. That is called as irreversible inhibition. In irreversible inhibition, the inhibitor bind covalently with the enzyme and inactivate them, which is irreversible. These inhibitors, irreversible inhibitors, are usually toxic or poisonous substances which destroy the activity of the enzyme. Now, these inhibitors, they generally form covalent bond, and we know covalent bonds are quite stable bonds, to an amino acid residue which are present near the active sites. They permanently inactivate the enzymes, and generally these inhibitors, irreversible inhibitors, they generally bind to the amino acid like serine or cysteine residue, because they have the reactive functional group, like serine has OH and cysteine has SH groups, which are quite reactive groups. So these inhibitors can attach to these OH and SH group and block the activity of the enzyme. But this blockage, it is a irreversible manner. That means these inhibitors permanently inactivate or poison the activity of the enzyme. Some examples of irreversible inhibitors we will discuss here. The first example is of the compound diisopropyl phosphofluoridate. Diisopropyl phosphofluoridate, which is also called as DIPF. It is a component of nerve gases. It reacts with serine amino acid residue, and irreversibly inhibit the enzyme and prevent the transmission of nerve impulses, which are really very essential to transfer the neuromessenger from brain to other part of the body. But if diisopropyl phosphofluoridate is there, which is a component of nerve gas, it reacts with serine residue and inhibit the activity of the enzyme. Generally, it binds to the enzyme like serine proteases, acetylcholine esterase, etc. Now here you can see that this DIPF, this acts as an inhibitor, and this is a chymotrypsin enzyme. Chymotrypsin, this is the active site of chymotrypsin, and DIPF, its structure is similar to the active site of this chymotrypsin. So it attack on the active site of the enzyme and it block the activity of the enzyme. So this DIP chymotrypsin, it becomes completely inactive. It is the example of irreversible inhibition. This process cannot get reversed. Another example is iodoacetamide. See here, this example you can see, this is the iodoacetamide. It modifies cysteine residue. This is the enzyme which has cysteine amino acid, and cysteines, they are having SH functional group. So this iodoacetamide, it modifies the cysteine residues and hence may be used as a diagnostic tool in determining whether one or more cysteine residues are required for enzyme activity or not. So in this way, here this iodoacetamide, it is acting as a irreversible inhibitor to determine about the nature of the cysteine residues present in the enzyme. Next example of this irreversible inhibition, it is the penicillin antibiotic. All of you know that penicillin is used for the treatment of bacteria. So the penicillin antibiotic, it acts as a irreversible inhibitor of serine containing enzymes and block the bacterial cell wall synthesis, and in this way it inhibit or deactivate the actions of the bacteria in the body. Next example of irreversible inhibition, which is really very useful, that is disulfiram. Disulfiram, which is also called as antabuse. Now, what do you mean by booze? Booze means it is a alcoholism. दारू पीने किंवा शराब पीना. Now, disulfiram, it is used as an antabuse drug and it is used in the treatment of alcoholism. What happens when this disulfiram is given to the person who has a habit of drinking alcohol? Then when disulfiram it enter into the body, it make the person quite sick. Why? Because when this disulfiram, it acts as an irreversible inhibitor, and what it does, it first acted upon one of the most important enzyme, that is alcohol dehydrogenase. Alcohol dehydrogenase, this is the enzyme which is present in our body. What it does, it converts alcohol into first acetaldehyde, and then acetaldehyde get converted into acetic acid. If this process is continued, then the person who is drinking alcohol, he cannot leave the alcohol easily, because alcohol is converted finally into acetic acid. But when disulfiram or antabuse, this is used, it acts as an irreversible inhibitor, and what it does, it attacks on the alcohol dehydrogenase enzyme, it inhibits its activity, and what happens, this enzyme, it converts alcohol to acetaldehyde only, and due to this, the accumulation of acetaldehyde in the body increases, and which make the person sick, and then that person who has a habit of alcohol drinking, it start avoiding the drinking of alcohol. So this is one of the important use of irreversible inhibitors. So till now we have discussed various type of inhibitions like reversible inhibition, irreversible inhibition. irreversible, competitive, uncompetitive and non-competitive inhibition. Here one table is there where we can see or we can compare these inhibitor. First is competitive inhibitor, but it is reversible. Competitive inhibitor, but this is irreversible, and then non-competitive inhibitors. So only three are taken: reversible, competitive reversible, irreversible and non-competitive inhibitors. Now we will see resemble substrate in competitive. Yes, substrate and inhibitor, they resemble with each other and hence they compete with each other to attack on the active site. In case of irreversible, yes, they resemble with each other, but in non-competitive inhibitor and substrate, they don't resemble with each other and hence there is no competition between them to attack on the active site. Achha, whether they attack on the or bind on the active site in competitive inhibitor, yes, they attack on the active site. Here also in irreversible, they attack on the active site, but non-competitive inhibitor, they don't attack on the active site, but they attack on the other site, that is called as allosteric site. Now what is the effect on the Vmax? In competitive inhibitor, we saw that Vmax remain unchanged but Km is increased. While in irreversible inhibitor, the Vmax is decreased but Km remains unchanged. While in non-competitive inhibitor, Vmax is decreased and Km remains unchanged. Now this inhibitor, can it be overcome by increasing the concentration of substrate? In first case, yes, it can be overcome by increasing the concentration of the substrate, but in other two cases, there is no effect of concentration of substrate on the inhibitors like irreversible and non-competitive inhibitors. Thank you so much.